The speaker, Dr. Shakir Jawad, works as a senior advisor on global health and provides high-level technical consultation in the field of biological containment, emerging infectious disease, especially dangerous pathogen, and of health concept of a number of American institutions and ministries of health in 7 countries outside USA. He will talk about vaccine types and merits. Dr. Shakir.
Thank you very much, Dr. Maitham, Dr. Jasm Al-Azawi, Professor Rawaf, Dr. Jawad, if he's here. Thank you very much for having me on this meeting, and I will share my screen now so that we can start.
Yes.
And I think it's visible now, correct?
Yeah, yes.
Okay. It's going mostly be like a visual presentation. Most of the slides I have are visual, are like kind of tables, pictures, animations that I created so that we can go through the process more quickly and easily. I try to simplify it as much as possible because probably not all the people who are attending are from the medical field, but I hope it's well understood and easy. So the views expressed here are those of the author. They do not represent the organization I work for or the US government, because we take funding from the US government. And it is not intended to be professional medical advice. You'll have to take your healthcare provider's opinion if you have anything. And this presentation is only for information. So let's look into what's going on in the world right now. It is how many people have been vaccinated so far. This is like an hour ago, and we have like almost 750 million people have already received 1 dose, at least of the vaccine, and that makes 9.8 doses for every 100 person in the world. So the process is going well, regardless. Like there is definitely a very high disparity between countries that are receiving those vaccines. Countries that are producing the vaccine, countries that are rich are getting more out of the vaccine. But in general, I think the process within that period of time is going well. We are very much progressing in the right direction. And despite all the negative comments and remarks done by so many people, but people are going towards vaccines whenever they are available. So the current status now is we have like almost 136 million people affected, and deaths are almost 3 million now. The problem what we are seeing right now is this trend. It's going up. Cases are now going up globally all over the world. That's in general, like it's an observation that has been growing, and people call it third wave, people call it fourth wave. It depends on how you describe it and where you are in the pandemic cycles. But let's look into different countries. This is daily new cases in Israel. And you can see there is this dramatic drop, and there is no surge. There is no increase in number of cases, and we will discuss why. And the next one is, sorry, this is the UK. This is Israel. This is the UK. And this is the United States. The United States has definitely dropped a lot, but now you can see there is some picking up of cases. It's going up over here, but compared to the world, it is not as bad as expected. There are increasing number of patients now, people getting more into the ERs and admitted to the hospitals, but it is still not as pronounced as it can be seen in Europe and other countries. This is Iraq. Unfortunately, Iraq now is recording cases more than it's ever had over the history of the problem of the disease of the pandemic in Iraq, even higher than the highest levels recorded in the past. Of course, you will see deaths following because usually deaths follow after 1 month. And that's a very alarming situation. Let's see why, and by comparing how much vaccines those countries are taking. This is the number of vaccinations taken per one vaccination doses, total number of doses. So you can see in Iraq and in the United States, sorry, we have 178.8 doses of vaccine given so far out of 330 million of the population. That's more than half, and you will see the percentages. In the UK, there is like 38 million.4, and in Israel, it is like 10.22 million. Israel's population is 9 million without the occupied territories and the West Bank and Gaza Strip. And in Iraq, let's see how much. We are only 103,000. We are one of the countries that with minimum like number of vaccinations all over the world. And if we look into the percentages of how much we are having, we can see that Israel is like 118 per 100. So it's past the total population and is going out and is revaccinating people more than the second dose. This is a percentage per 100 of population. And if you look into the United Kingdom, 56.6%, the United States is 53.47%, in Iraq it is still 0.26%. That's less than 1%, it's a 0.25%. That will show you why those figures I showed you in the beginning of the previous slides, why they were like climbing up in Iraq and not in those countries. Actually, they were going down. The UK is almost already put an exit strategy from the pandemic by declaring the next step. Hopefully they can do that, of course. So let's look into the vaccines, where we are in the vaccines globally. There are 77 vaccines now in the animal preclinical testing phase. And we have 86 vaccines in human trials. Those are distributed over here. Of course, if you like put the numbers together, you can see there are more than 86. This is because like they are doing phase 2 and phase 3 at the same time. That's why you can see the same drug repeated more than once, and that's why they show those figures higher than usual. But anyway, those are what we have right now all over the world, vaccines. We have 50 in phase 1, which is done on 100 people, 35 in phase 2, which is done around 1,000 people, and 23 in phase 3, which is done on tens of thousands of people. We have 5 authorized, 8 approved, and 4 abandoned. They were refused. So bad vaccines will not continue to be used. People who are talking about like AstraZeneca vaccine and that it's made to kill people, that's not like, this is nonsense. This is stupid statements done by some stupid people. So let's look into, this is just a demonstration how the vaccine works against the virus. This is coronavirus. Usually it gets to the body through ACE2 receptors. Those are ACE2 receptors, angiotensin converting enzyme 2 receptors. And this will, sorry, someone sent me a request. And this will go in, and once it goes inside the human cell, usually cells are all over the body that has those ACE2 receptors. You can find them in type 2 cells in the lung. You can find them in the nervous system and inside blood vessels, inside the heart, the cardiovascular system, kidneys. That's why the disease is all over the body. It affects many systems. They will immediately go for opening their core and get the genetic material, which is RNA, to the ribosomes. So virus genetic material will go to this R. R is a ribosome, which protein synthesis, where protein synthesis takes place. And then it works like a copying machine. It keeps on copying material, genetic material of the virus. The virus uses the machinery of the cell to reproduce itself. And this is how the disease gets inside our cells. And once it's inside the cell, it's very hard to reach by medicines and drugs and medications. Then the virus will form and it will keep on going out. And this is what we call viral shedding. And the more we shed, the more viral load we have. And that's going to be measured by number of those viruses in a millimeter of blood. So viral load is an indicator of how severe the disease is and how patients are responding to treatment once they get. So those are the like immune system products. We have antibodies and we have T cells. Those will jump around and be ready to go and attack the virus itself once it gets into the body, and they will change the shape of the spikes. The spikes are what the virus uses to get inside the cell. So when it comes to get in, it is not going to be able to admit itself or stick itself to the ACE2 receptors. Those variants, this is the V's for variants, is when the spike changes in a way that they will not anymore resemble the older structure, and the antibodies will fail to recognize them, either partially or completely. Right now they can, but it's partially not able. That's why there is some kind of less potency for some vaccines to attack some of the variants, not all of them. And the variant can evade or avoid those antibodies and can come back and get in. That's why we are in a race. We're trying to get people as much as possible vaccinated before we reach to a variant that's 100% avoiding the vaccine antibodies and the T-cell immunity. And then we end up like with a new cycle of vaccination, which is endless. Those are the two main variants that are for us considered like of concern. The Kent or the UK is the most widely abundant variant that's now going all over the world. And this is the most dangerous one, the South Africa, the B1351. This is the most dangerous one because it almost neutralized AstraZeneca vaccine and it's not effective against it. That is what's happening right now. So we are in this race, but still many of the vaccines are still effective against it. So let's look into why do we vaccinate? We vaccinate so that we can reach the herd immunity or community immunity, which is the right term, the most appropriate term. And we can reach it through vaccination, which is the safest way. We can reach it through infection, of course, but that's very unsafe, and millions of people will die until we reach in the world to this type of immunity. The reproduction rate is very much determining how much people we need to vaccinate. Coronavirus infection rate worldwide is 2 in 100. Like each person will infect one infection rate. Like one person infects 2. But in the United States, it's 9 in 100. Like that's the difference between the two countries. And if we look into all those older diseases that are very well established, and we are controlling them, not because they disappeared, we only are controlling them because of herd immunity, like diphtheria, measles, mumps, pertussis, polio, rubella, and smallpox. And you can see the herd immunity threshold of vaccination. We need to vaccinate those percentages of people. And the more we get transmissibility of the disease, the more, like in here, the more transmissibility is there, the more is the number you will see here. That's why we jumped from 60% when we started looking into coronavirus, and now we are looking into 85, maybe higher percentage of vaccine coverage or people, immune people, so that we can get the herd immunity. So vaccines usually here start like an initial exposure, or probably this is the disease, primary immune response takes place. And then when second time you're exposed, the response is going to be very robust and strong, and it will be able to control the disease immediately. So you will not have symptoms or might have mild symptoms, but you will not go to the critical and the death phases of the disease, hopefully, if you have a normal immune system. And this is how they go, like by IgG, IgM. IgG is later, the blue one. IgM is always earlier and is less. So this is not very important. The immune system, we have the innate immune system, this one, which gets weaker with age. The older we go, this gets weaker. That's why older people get more disease, worse disease or severe disease compared to the other people, younger people. And we have this one is the adaptive immune system, which gets stronger with time. It gets educated. Each time you're exposed to a pathogen, this one gets educated and creates adaptive immunity to that. And that's what we want. And if you look into the cells here, memory T cells, cytotoxic, the helper, and the memory B cells, those are actually the cells we the main cells of the excuse me, of the human immune system. So we have the memory B cells, antibodies, and the killer B cells and the helper T cells. Sorry, killer and helper T cells, CD8 and CD4. And this is what they attack. Usually the memory B cells will attack the binding domains at the spike protein, the two main proteins that are antigenic to us and are recognized by the immune system are the spikes and the nucleocapsid, and that's the cover that covers the nuclear material inside the virus. And this is how the T cells attack the virus itself as a whole. And we go back to this. So let's see what types of vaccines do we have. I had to go through that introduction just to make you understand how those vaccines will work. So we have four types of vaccines. Of course, there is a fifth one that is the TB vaccine that is still under experimental studies by the Murdoch Institute in Australia, but it is not anymore progressing anywhere. But we have four vaccine types of vaccines. I'm talking about categories, not by name. We have the viral vector vaccines and the genetic vaccines, and those uses body machinery to develop the epitope. The epitope is the antigenic part of the vaccine that the immune system will recognize. And we have protein-based vaccines and attenuated or deactivated vaccines, which are like an epitope is ready to use. We put it inside, so the body is not going to make anything. It will immediately attack whatever we are getting in. The first two are more complex, and the second two are easier and older, like older fashioned like vaccines. Let's look into those. First, the viral vector vaccines. Examples are the AstraZeneca and the Sputnik V, the Russian, and Johnson & Johnson and CanSino Bio. Those are from China, CanSino Bio. And this has vaccinated millions of soldiers in vaccine in the beginning of the whole story. So those, how do they work? If we take the viral vector vaccines and look at them, they use adenovirus, DNA, which are DNA vaccines, so they use the adenovirus. And if you look into adenoviruses, this is the replicating segment. In the United Kingdom, they used chimpanzee adenovirus, so it will not cause disease in humans, and the human body is not going to recognize it because probably you have some immunity against human adenovirus from getting common cold, because it's one of the diseases that causes cold in winter. So they used the adenovirus. They removed this replicating segment. This was removed from the adenovirus, and they replaced it with SARS-CoV-2 spike protein replicating segment. So when they put this inside the body, this segment will create a spike protein, the spikes of the coronavirus. So let's see what's going to happen. So when this gets in, this is a model of a cell, a very simple model. Because this is a DNA virus, it needs to transcript into messenger RNA and then go to the ribosome here so that it can synthesize the spikes, the protein that we need to energize the immune system against it. And a lot of people think this is dangerous, it goes inside, but the way this was designed and the lack of binding enzymes, there are no binding enzymes, and there are stop codons, and the ends, the free ends of the DNA are managed in a way, this is all recombinant technology, so this is all like done in a lab, are not like free open, and they do not match any of our nucleotides, and that's why it is not going to mix with our DNA, and it's not going to change our genetic material. So this will transcript into two mRNAs, and then it will form the S protein. This is the spike protein or the S protein. And usually once it's synthesized, it goes out to be like displayed on the outer part of the cell. And of course, the immune cell system will send antibodies and T cells to recognize it and fight it and neutralize it. That is those are how AstraZeneca vaccine, which is present in Iraq, is It is a safe vaccine, and I will show you numbers then by the end of its safety. And the next type of vaccine is the genetic vaccines. People kept on talking on this from the beginning. Pfizer and Moderna are the most popular too. Of course, there are like kind of 18 another ones in progress right now, and different levels of studies. Those actually people kept on accusing them of they will cause some kind of genetic changes in our genetic material. But actually, this is nonsense because those will never go once they are injected. This is a sequence of genes, nucleotides put on a strand and a single strand because it's an mRNA, and it will go only inside the cytoplasm. It's not going anywhere near the nucleus, the DNA, and it's not going to be, although the previous one goes inside, but it is very well protected by design. And once they go in, they immediately will work with the ribosome to create the S protein, will go out, and then it will be recognized by the immune system. So if you notice, this one is one step less. The first one will send a DNA that needs to decouple inside a transcript inside the nucleus and then go outside as mRNA. This one is immediately putting an mRNA. They don't need to like decouple the DNA double strands into a single strand anymore. If we go to the next type, protein-based vaccines, the most popular two, none of them have been approved yet, but we have like Rocket from Russia. Rocket is actually, Victor. Victor from Russia is being now used like some kind of, it's only like still phase 2, just entered phase 3, but it's used in Russia without much precautions. And Novavax from University of Maryland, United States, and this one is phase 3, and it's showing very good and promising outcomes. This one looks good. Those are done this way. If this is the virus and those are the spikes, of course, this has been scaled so to demonstrate to you in a simplified way what's happening. So they keep only the spike. Let me go back and show you. Look at this one. This is the only thing that is kept from the virus, only the spikes. And they are complete and ready and physically available. Those spikes will be, of course, treated by adjuvants and nanobodies to protect them and make them more like livable inside the human body. And then they will be injected. And then the antigen presenting cells will attack them. Once they go in, they will be attacked by the antigen presenting cells. They engulf them, look at them, and then they present them to the outside. Once they are presented to the outside, the immune system will immediately send its like fighting elements to neutralize this new pathogen that is recognized. That is how this vaccine works. The last vaccine is the inactivated or attenuated vaccines. There is no attenuated vaccine for corona yet. There's one being developed in Los Angeles, and it's a nasal spray, and it has not yet like gone. So every patient is safe. No one is going to get the disease from the vaccine because those vaccines are not attenuated. They don't have viruses inside of them. They only have inactivated or parts or recombinant products. So the inactivated virus, this is coronavirus. Now it's activated. I changed it from colored to black and white, so you know it's inactivated. And then this is going to be treated, of course, with all the adjuvants and the technology to protect the virus, the dead or killed. We don't say killed because viruses are not living creatures. It's a wrong term. Viruses are a chemical structure, nucleotides and strands and some like protein around them, but they are not living. They only replicate inside cells. That's the term, replication. They don't live. So saying killing is not a right word, but it's a common misnomer used always. So we better say inactivated viruses. So those inactivated viruses, once injected inside the body, again, the antigen presenting cells will attack them and they will present them outside. The good thing in those vaccines, theoretically, is that they have multiple antibodies and T cells, different colors, if you notice. That is what we call a multiple epitope vaccine, a multi-epitope vaccine. A multi-epitope vaccine is a vaccine that can attack every element of the virus. Those are the epitopes of the virus. We have the spike, the membrane, the small envelope, the nuclear capsid. The main two ones that are responding or showing antigenicity in humans for the coronavirus are the spike and the nuclear capsid. So multi-epitope vaccines are supposed to be like, they can fight those variants that are happening, because mutations are mainly happening here. And we have like a new strain or variant coming that's probably also affecting the nuclear capsid, and people are concerned about it because this means there will be no immunity at all for the vaccines. So if you look into those experiments, this is the S protein and this is the nuclear capsid proteins. This dark color shows reaction to the antigen, like to the epitope. And that means like the vaccine is fighting both of them. And this is the value of having the old traditional killed vaccines or inactivated vaccines. They are more diverse and the range they can fight the virus through. So those are the main epitopes of concern. We have the United Kingdom, South Africa, Brazil, and California. And this D614G looks like is the common denominator for all of them. They all have this mutation, which is now like getting more attention that it's probably due to this common mutation that are spreading faster, only about spread. And still you'll have to wear a mask to get rid of them and apply public health. Those are the vaccines that were abandoned. They were not used. Imperial College had to stop working on its vaccine, and they're looking into developing vaccines towards the variants, because they thought like the market is now full of so many things and we cannot probably compete in the right time. Merck, which is a huge American company, it's a multinational, of course, and worked with the Institute Pasteur in France, and with IAVI, which is an NGO, like works on HIV vaccines very much. And they worked with them. Both of them could not provide enough immunogenic reaction in the vaccines they provided. So they stopped, they were abandoned. And Queensland University of Queensland in Australia used like in their preparation a protein that unfortunately made patients show positive for HIV disease, HIV virus. And that was going, although they are not having the disease itself, but this will very much distort the epidemiology measurement of the disease. And those people will mix with the patients, and it's very hard now to tell which one is a diseased patient and which one is from the vaccine. So they stopped it because it's not. So but vaccines, if are not good, they will stop. Millions of dollars have been spent on those. And probably Dr. Waf can tell us how much Imperial College spent, but this is the thing. Now Pfizer requested FDA to approve use of COVID vaccine in kids aged 12 to 15. They finished their studies and it's 100% effective. If you see the number here, 100% effective for children. And I saw today an FDA release that they will look at it very quickly. So hopefully there will be more. And Pfizer also began COVID vaccine trials in younger children, 6 months to 2 years, 2 to 5 years, 5 to 11 years. So I think by the end of this year, all school children will be vaccinated. They can go, that's the United States. They can go inside universities and schools, and that will be much safer. And it's going to be a very good experiment. People who say vaccines are not good, look at this. This is the non-vaccinated, or those who have the placebo, compared to those who have the vaccine. And look at the percentage of how many people have been hit by the disease here and how many people were able to avoid the disease here. It's impressive, very impressive. And always vaccines and medicines and everything else are under this equation, risk or benefit. If benefits are higher, way higher, then we go for it. And how long vaccine-induced immunity works? So we have like two types of studies: the in vitro studies, which Iraqis are very interested in, and some people also, some places in the world, IgG, IgM, IgA, cellular response, they measure them and they think that's the only way they look at it. Actually, the best way to measure vaccine effectiveness is population-based observational studies. Like you have to observe people after they take the vaccine, and then you can take the effectiveness of the vaccine. That is very important. So those are like the studies that have been done lately about the antibody status and how can people can be safe and whether they get reinfected. SARS-CoV-2 reinfection can occur, but it is a rare phenomenon. Protection against repeat infection was 80.5 to 95%. That is how much, of course, in the older age after 65, it gets less, but it is very effective. Like reinfection after vaccine or after the disease is not common. And there are two terms, of course, we always use, efficacy and effectiveness. Efficacy is what we measure in clinical trials. For example, Pfizer protects like only 5% infected after the vaccine. That's a clinical trial efficacy. But in a new Israeli study that was done on 700,000 fully vaccinated people and were thrown into the community effectiveness was 0.04 only got infected. So it's much better than the 5%. So we need to be careful on using the two words like efficacy and effectiveness. Each one has a different meaning. This one is for clinical trials and this one is for a true community and population-based studies. We need to stop replication because replication of viruses means mutation every time there is a replication process. This is how. We need to cut the chain of infection. There will be no more infected people. There will be no replication by public health measures. Only like masks and vaccines, of course, work very well, but even masks and social or physical distancing and not attending groups. Replication, if we stop replication, we stop mutation, there are no new variants and we are safe. That's very important. Stop the replication by stopping the transmission. So I'm almost done. This is like the AstraZeneca latest study, like there were 25 million doses given and there were 18 deaths, and that's the percentage of the deaths compared to the number of doses, and that is like 0.27 million percent. Sorry, per 1 million, there's less than 1 per 1 million. Those are like the general outline of the safety of the, of course, there's a new study, two new studies actually that were released about AstraZeneca, talking about platelet factor 4, vaccine-induced immune thrombotic thrombocytopenia, and linked to heparin-induced thrombocytopenia. These are all like the studies that are going on. That's my picture taking my vaccine in a car. And this is how I got my vaccine because I was in a hurry and I wanted to take it as early as possible. And I went to those places that vaccinate 500 vaccines per hour, and they work for 10 hours. And we have so many of them in my state, and that's like 5,000 a day, only this center was doing. And you can imagine why the United States is vaccinating 4 million people per day now, almost. That's still valid. Masks, distancing, avoid gatherings, don't travel, and those are my references, and thank you very much. I'm sorry if I take too much time. Thank you.
Okay, thank you so much, Dr. Shakir. Now